So first of all, it's a pleasure to speak here, and I will try to reach the top bar that we got this morning from the excellent talks. I'm sure I will not reach, but I will try at least. So I'm going to talk a bit about one specific aspect of what we do. But first, I wanted to say, yes, as all of us, or most of us, we have a very international experience. So I did my PhD in Valencia. And then following the good weather, I moved to Germany. And now I'm in Stockholm. And I'm really happy there. So I have been there since 2016. And my laboratory interest combines two things, basic molecular biology and also the development of new technologies for biomedicine. So one part is the study of RNA life from transcription to decay. I will explain a bit what it is this, don't worry. And then we also have a lot of work, let's say in cell-to-cell variability, cancer plasticity, and aspects like that I will not go into detail. And of course also development of new tools for diagnosis from cancer to SARS-CoV-2 and these kind of things. So I just decided to choose one specific topic, and that's the gene expression, and I'm going to talk about RNA. But what is RNA? So you all probably know, with probably one of the aspects of this pandemic is that everyone knows nowadays what is RNA and what is a PCR. So that will be very useful for this talk. But just to keep it short, we have the information. It's in the DNA. Here it's in eukaryotes. We have a chromatin that is kind of an organization, and this information has to be copied to the DNA, to the RNA. And this RNA, there is a process that translates this information in this RNA into proteins in the ribosomes. So I'm going to talk about how this flow of information, at some moment, it has to change. So when you have a molecule of RNA that produces proteins, that is used to produce proteins, the cell has also to remove it. And that we do by RNA degradation. And I'm going to discuss how this process, that we thought they were very different, are in fact not so different, or they are more close to each other. And I'm also going to talk about a revolution that I had the pleasure to experience during my scientific life. When I was a student, we were still in the individual gene analysis, where you have a method where you study one specific aspect, one specific gene, in this case an Northern blot, or you do kind of like where we do a PCR, you look for a specific gene. But then with use of technologies from semiconductors and so on, we were able to multiplex and get many more information at the same time. And there was something that was very popular 15 years ago, that was DNA microarrays technologies. And in the last 10 years, we now are able to have a thing that we call it sequencing. So basically what this means is that now we are starting to get data. We are not at the level of a physicist, but we are trying to go into that direction, to get a lot of data to be able to interpret. And my lab focusing in this. And this also allows us to make questions that we could not do before. So I'm going to talk about a kind of molecules that are the 5' phosphate molecules. So just let me say you have a molecule of an RNA. You can imagine like a string. And on the top you have a blue one, and we have a structure that is a cap, that is, imagine like a cap that protects one side of the molecule, and then you have this ACE at the other side that protects the other side. And this is the molecules that normally we studied this is in bad yeast, for example, or humans. And during my studies we decided to look not only those molecules that are the standard ones, but the molecules that have lost this cap and are being degraded by the cell. And that was normally considered, okay, this is something that is getting rid of. So we just controlled to see what was happening there. So this was a work that I did together with my colleague Veivu. This was at the end of my postdoctoral stay at EMBL in Heidelberg, or Stanford, because our lab was in between both. And basically, I'm just going to skip this talks, so these plots, because are too specific. But you can see below there is this kind of lines. And just to see this red bar here, this is the region of the DNA that has the information for this protein, that we call it BAT2. If we look only to the end of the molecules, these are the normal molecules that start before and then continue. And then we have these molecules here that don't have this protection cap, and they are in the body of the gene. They are all over the gene. So initially we thought, okay, this is just noise, who cares? This is not important. But when you analyze it in more detail, we saw something that was very surprising. We took all these boundaries and saw that in fact they were a periodicity. There was a theme in them. We had a periodicity every 3 nucleotides, every 3 letters, let's say, of the genetic code. And 3 letters are very important in biology because that's basically the basis of the genetic code, where you change the information that we have in the DNA to the RNA and then the proteins. And we saw this pattern that basically we saw that every 3 letters we had kind of a peak and valley. And that was very surprising. As I was telling you, these are molecules that are being degraded by the cell, that they are disappearing. So what we realized, there is different ways to get these molecules, but initially we thought that the molecules that are being removed from the cell, they are not used anymore to produce proteins. But our work showed that in fact those processes were happening at the same time. So now we know that these 2 events happen at the same time, and that tells us 2 things: how this is happening in the cell, and that's important, but also that there is information in the cell that we were not using before and now we can use. So we are trying to use this information to learn about biology. So these are just to show that what we saw is true. We did our controls, and I'm not going to go into detail, it's too technical. But we understand in budding yeast, the yeast that we use to make wine, for example, how it's happening, and we know, let's say, this Pac-Man that follows the ribosome can really get a nice interface, and we can disposition. This is a nice example where we decided to say, okay, what happens if we make these machines that produce proteins stop in a specific position? So here we have the cells, all the genes, and this is histidine, that is one of the amino acids. And we have cells growing in a normal media, in a complete media, and we have this periodicity every 3 nucleotides. Then we grow cells in a media that don't have this amino acid, so the cells have to produce it. So it becomes limiting, and this machine that is copying needs to wait a bit because it needs to arrive a new molecule. And then we see that this molecule was waiting here, and that was enough for the Pac-Man to reach it and make this peak. So basically this is the peak that the Pac-Man is making because there is a ribosome slope here. And when we really increase this, because we make that there is not more of this amino acid, this peak was so big that all these machines start to collide with each other. So this was just a proof that we really are able to see by studying what is in the cell, what's happening, basically. So I'm just going to jump, but just to say that now we have a method that is very, very easy because we don't do the experiment. The cell does the experiment for us, and we are just kind of digging into the trash of the cell to learn what was happening before. And this opens us a question, and one thing is, okay, we know this happening in yeast, we know it happening in plants, we have some evidences that it happens in humans, although it's more complex. And we have many tools that we have developed in my lab and other labs to understand this, computational and experimental. But this offers something interesting. As we don't need to do an experiment and the cells do the experiment for us, can we use this to understand the microbiome? What is the microbiome? Probably many of you, it's the first time you listen to this word. So you all think that you are all humans, and we are all humans. And that's true, we are humans, but we are also covered inside and outside by a lot of bacteria and different microorganisms. And in fact, the microorganisms living on top of us or inside us, they are essential to maintain our health. And we know that variations in this microorganism can be associated to diseases or different metabolic problems and so on. So it's important to understand how they are and how they are unbalanced or how they respond when we take a drug or antibiotic. So we know it's very complicated, but we know that they are essential to understand life. So we decided to see if this was happening in prokaryotes, that are this kind of more simple organism. But simple organism is not a good term, because in fact they are extremely complicated and extremely variable. I'm not going to go into this, but this is just to show that what we know of how this RNA disappears in the cells in eukaryotes is very simple compared to the diversity between the different bacteria species. And quick answer is that the same thing we try to answer. Okay, if we can make this bacteria to tell us how it works, maybe we can learn out of it. The answer is that it works. And we can now, this is an example with B. subtilis, that is a model organism, that is a bacteria growing in the ground, and we can see that we can follow the path, and we can tell, without doing anything, how were these ribosome producing proteins in the cell. And we can also use this when we treat with some drugs. We can see how these proteins, how these ribosomes change position. There was, of course, the work of two talented postdocs in my lab, Susan and Lilith, that they did the actual work. And then just to show you that this happens in many species. So this is different lactobacillus, for example, that we use for making yogurt. But in other species, like for example E. coli, that's not the case, but that's because they have different machinery to remove the RNA. But what's interesting that even if we don't see this specific interaction that I was showing you before, we still are able to see variations in this trash of the cell that we can use. And we are trying to use that to understand how we can get a very fast response to drugs. There are some examples of cyanobacteria or different species. But the only, just to start to finish, the only thing that I wanted to show you is that we can use this kind of very leftover of the RNA to understand how cells are responding to the environment or to drug treatments. And this is really fast because the cells don't need to produce more RNA. So one application that we are extremely excited is that now we can use this to look for ultra-fast drug responses, because basically we expose a drug to the cell, and if the cell responds really, really fast, we'll see a difference, because basically there is no need to produce an RNA, there is no need to read the DNA, produce an RNA, produce a protein. You just see this, movement of the ribosome. There is a small change, and as we are able to look the trash of the cell, like you can this trash tell us what is happening right now. So this is really, really nice. And another thing that is important is that this method give us kind of very long reads, long stretches of letters, and this makes us very easy to distinguish different organisms. So in this case, I'm just showing one example where we have two lactobacillus that we mix in very controlled situations, and we can see that even if one is at 0.01%, we can tell it very easily from each other. Of course, this also works in patients. So this is some samples from bacteria from vaginal swabs. We have done also with feces from patients or different things. So we can tell how were the bacteria in this case, in these women, behaving at that moment when the sample was collected. And the next thing is that we did not need to do anything. So with this, I just wanted to, I mean, this is a bit too specific for this audience, I'm sorry, but just to say that there is information even when you think that there is no information. So it's good to develop new methods that allow you to look in a slightly different way. So sometimes it's good to be a bit naive of what you know, because then you can ask questions that maybe is stupid or maybe not. And sometimes you can learn new things. And now we are trying to use the medical application of this approach mainly to understand how drugs interact. With that, I just want to thank my team, all the collaborators, and of course the funding and all the organization. Thank you.